everyone. My name is Alison Devault, and I'm the Director of Genomics at Daicel Arbor Biosciences. I wish we could have traveled to be there in person at the ConsGen meeting with you, but I'm very pleased to be speaking with you today and that Arbor had the opportunity to be a sponsor for this great meeting. The topic I will be speaking about today is highlighting how hybridization capture technology, specifically Arbor's line of myBaits target enrichment kits, can be deployed as a powerful tool for efficient and effective data generation for conservation genetics applications. myBaits has been embraced by researchers studying phylogenetics and population genetics across a wide variety of organisms because it enables many of those core benefits of next generation sequencing, or NGS, while maximizing sequence efficiency. In today's talk, I will start by reviewing where the hybridization capture technology falls in the modern conservation genetics toolkit. I will highlight a couple interesting case studies in which researchers utilized myBaits for conservation genetics research. And finally, I'll follow up with a deeper dive into the mechanics of Hyb capture and how its molecular underpinnings by nature make it a flexible and versatile technique. The landscape today for techniques that are available for modern conservation genetics is, as I'm sure you are all very well aware, maybe painfully, it's full of options. So there are benefits and caveats to all these approaches, depending on factors such as how well sequenced your organism of interest is, what is the breadth of your reference database, what is the quality and quantity of genetic material that's available for study, are you working with mixed source or contaminated substrates? What's the scale of your project, etc. So many variables, and all of these can make choosing one or more techniques for a specific project pretty complicated, especially for projects that involve a range of sample types or sources of DNA, particularly things like working with environmental or museum specimens in addition to fresh specimens. So today, I'm going to be focusing on how NGS paired with hybridization capture is a strategy that is particularly well suited to meeting those particular needs and challenges that are found in conservation genetics projects. But of course, that doesn't mean that all of these other options are also not very powerful in their own right. And indeed, Arbor routinely works with and supports researchers who are using multiple techniques across their research programs, or even within the lifetime of a given project. So to help break down all those pros and cons, I've compiled this table, which attempts to score these various techniques on a range of attributes, particularly relevant to a conservation genetics researcher during their decision-making process. Try to rank these in as fair a manner as possible, considering typical usage, but of course that's tough since most of these are going to be affected by project scale and sample-specific factors, so got to keep that in mind. That being said, some of the more traditional genotyping methods, such as STRs or microsats, or SNP chips, those of course can be very powerful and very cost effective, particularly for routine screening or large sample numbers. But they do require a good deal of prior information about the target genome in order to design the primers or the probes, and they do have limited to no discovery power for novel variants or mutations outside of a very narrow scope within their target space. Amplicons or individual PCRs paired with Sanger sequencing can also be very powerful, but again may require a lot of upfront work and knowledge for primer design, and you have to have sufficient reference databases, particularly for multi-species panels, and they are also not suited to variant discovery for all types of mutations. If we consider purely the total scope of data that can be recovered in a single assay, the gold standard right now is still total shotgun, total whole genome sequencing, where we sequence all the DNA in a sample using next generation sequencing. This is the least biased and the most comprehensive way to survey all genetic content in any type of sample. And assuming that you've selected the right library prep method and the right sequencing platform, short or long read, you in theory should be able to study any known or novel variant, any type of mutation with high breadth of data. But of course, as we all know, the downside of that could be that it can come at significant cost per sample, particularly if the genetic regions of interest or molecules with informative signal for your given research project are relatively rare within the sample. Worst case scenario being mixed source samples like environmental DNA, museum DNA, ancient DNA, where those samples are containing DNA from multiple sources, endogenous and exogenous, which based on the fact that many projects, you need to survey many samples, this really puts deep whole genome sequencing really out of the running, simply due to the cost. Some alternative approaches that would have relatively unbiased discovery potential while reducing that total sequencing effort required would be things like low-pass whole genome sequencing or shallow sequencing, or reduced representation techniques like RADseq. And those can be employed very effectively for some projects to lower that cost per sample. But both of those methods do in their own ways have pretty constrained variant discovery potential based on the regions that are being sequenced, and neither are particularly well suited at all for mixed source or highly complex DNA samples. So that's a limitation there. Again, going back to amplicons, that can be very effectively paired with NGS from one or more amplicons as a targeted sequencing technique, and that does allow highly specific amplification, and that can be very well deployed in mixed source samples. But again, amplicons, because of the nature of primer design, needing to pair, at minimum 2 functional primers together to get an amplicon in size and sequence, that can be somewhat restrictive to the discovery potential and the types of mutations that you're able to sequence. So finally, of course, there's the technique known as hybridization capture, which I'll focus on today. When deployed effectively, hybridization capture can basically allow you to access many of those best features of deep sequencing in terms of the breadth, variant potential, and mutations that you're able to sequence, compatibility with mixed source samples, but can lower that cost per sample of sequencing to allow you to deploy that targeted sequencing technique on a larger number of samples. And that can make it quite a comprehensive yet cost-effective technique appropriate for many conservation genetics type projects. So next I'm going to highlight a few studies that have used myBaits hybridization capture in research that have important implications for conservation genetics of their respective organisms. Just, because of the nature of time limits, I'm only able to mention a few of these today, but there are many more fascinating studies relevant to conservation genomics, phylogenetics, ancient DNA, all of these great fields that have used myBaits that I simply do not have time to highlight today. So I do encourage you, we have on our website a publications page where we do try to stay up to date on all these great studies that use our products. So the first study I'm going to highlight today is a 2018 paper from Aylward and colleagues in Ecology and Evolution. They used myBaits enrichment to recover mitochondrial DNA sequences from trace environmental DNA samples in their aim to study population genetics of the ii, which is an endangered lemur species in Madagascar. They looked to non-invasive sampling, specifically of wood samples collected where an animal, the ii, had previously chewed into trees to feed on the insect larvae inside. So the researchers took very small wood samples directly from these feeding locations that they found. They took them, extracted DNA from the exterior of the wood samples, and were able to apply myBaits' whole mitochondrial enrichment panel to pull down and reconstruct partial or full mitogenome sequences from almost all the samples that they tried in this manner. They were able to use these data for population level analyses in comparison to other sampling locations in Madagascar. And for some of the samples, they had also tried shotgun sequencing or total DNA sequencing. And in those cases where they have both shotgun and enriched data from the same sample, myBaits greatly increased their ability to observe the mitochondrial data by orders of magnitude in all of those samples where they had both data sets. This reinforces that hybridization capture can be a key method for being able to access extremely low signals from non-invasive sources of DNA, which are relevant for conservation genetics, like like environmental DNA samples. The second study I'm highlighting is in plants. This 2021 paper from Slimp and colleagues published in Applications in Plant Sciences, and they utilized a universal panel designed to enrich hundreds of genes from herbarium specimens from any angiosperm or flowering plant species. In this particular study, they were looking at a special collection of herbarium specimens that were collected in the 70s from Guadalupe Mountains National Park, which is in the US in the state of Texas. And they aimed to investigate whether this universal panel known as Angiosperms 353, originally designed more for broader scale phylogenetics work, if that panel could be applied and used for finer scale population genetics within different species of angiosperms. So for this study, they extracted DNA from all the herbarium specimens and performed enrichment with the Angiosperms 353 myBaits panel on all specimens representing 24 unique species with 4 samples each across angiosperm groups such as trees, shrubs, and grasses. For almost all the species, despite many of them being quite degraded, the DNA in those samples being quite degraded, they recovered nearly 200 or more kb of coding region sequence, which was directly targeted by the panel, as well as often the same or nearly even more sequence from regions that were of non-coding DNA that are physically adjacent to the coding regions targeted by the angiosperms panel. After their filtering for all species, they were able to retain sufficient levels of SNPs to calculate relevant population genetic statistics, even though they only had 4 samples per species. This is a great demonstration of the power of this type of panel, and the authors emphasize that these universal HybCap tools, such as Angiosperms 353, are very important research resources for investigating genetic diversity in wild populations through time and space, since they facilitate shared research on common sets of markers that are applicable across a broad range of species and can be used on a wide range of sample types, both fresh and, in this study, degraded material from herbarium specimens. The final study that I'm highlighting is a publication from this year by Cairns and colleagues in molecular ecology. The nightingale reed warblers are a group of birds in the Mariana Islands in the Pacific, in which all but one of the species has gone extinct since the 60s, and the only remaining species is critically endangered. In order to study genetic diversity of both the remaining extant species as well as the 3 extinct species, they extracted DNA from toe pads from museum specimens and feathers collected during banding of modern specimens, plus a few blood samples, extracted DNA from those and enriched with both nuclear and mitochondrial myBaits panels. They had a custom nuclear panel in the study which targeted almost 9,000 known SNP markers alongside specific genes that putatively coded for traits such as morphology and disease response. The data they collected from the samples allowed them to perform detailed analyses on genetic diversity and make estimates about features relevant to conservation in nightingale reed warblers, such as the population structure and historical population sizes. And the authors describe in detail the implications of their findings to ongoing conservation efforts for the remaining nightingale reed warbler species. Once again, this type of study highlights the great utility of myBaits hybridization capture approaches to enable detailed genetic diversity studies even in highly unique species and across multiple sample types. So with papers like these, I hope it's clear that myBaits HybCap can have great utility for conservation genetic studies. But you may be wondering, well, what is it specifically about hybridization capture that lends itself so well to a broad range of applications in this research area? So to that end, I will spend a little more time reviewing how the process itself works and why its unique molecular features are so advantageous for targeted sequencing in these applications. So to start, here across the top, we have a linear depiction of the typical next generation sequencing or NGS workflow, regardless of what kit or platform you're using. So whether you're doing short read sequencing, for example, Illumina, long read sequencing, for example, with Oxford Nanopore, this is the general procedure that would be followed in all cases. So you have your sample, you're extracting the DNA or the RNA in some cases, converting those DNA or RNAs into a barcoded library that is specific for each sample and allows it to be deconvoluted post-sequencing. And then prior to sequencing itself, you're applying this myBaits hybridization capture procedure. In that procedure, the library, again, it's already barcoded, is denatured or melted and allowed to hybridize to a complex pool of synthetic biotinylated oligos known as baits or probes that are complementary to the library molecules that you want to enrich. Once these complementary pairs have annealed to each other, that pair is bound to a streptavidin-coated magnetic bead, which grabs onto the biotin, and all the non-bound library molecules that are not of interest, so background DNA, off-target DNA, these are washed away, physically washed away. And then the now single-stranded target library molecules are eluted and amplified with universal primers and sequenced. At Daicel Arbor Biosciences, we have a unique oligosynthesis platform that allows us to build these biotinylated pools of custom oligos at any scale for any sequence. And these are provided in a kit, a myBaits kit, which includes the probes themselves, as well as all the reagents for the actual hybridization capture process that I just described. The key to all the benefits of Hyb capture that I mentioned earlier in this talk really lies in this ability for the unique target probes to hybridize to the library molecules that are complementary to them, but not modify them. And which means really two key principles are relevant here. The molecules of any length can be enriched with a given probe. So probes don't limit the molecule length that you can recover, which means that the same bait set can be used for both short read sequencing or long read sequencing, or both, and with libraries derived from any DNA quality. So whether they're very naturally sheared like ancient DNA or museum DNA or long, they can both be captured with the same bait set. And because these enrichment probes themselves are relatively long, at least compared to something like a primer, the minimum length for a HybCap probe that we sell is typically 80 mer at minimum. This allows that there can actually be multiple mismatches present in between the probe and the library molecule that it is pulling down. And enrichment can still function well despite there being some dissimilarity in the molecules that it is enriching. So those two fundamental principles, the probes themselves are independent of the library molecule, and that those probes can tolerate dissimilarity, really drive much of the ability and power of HybCap for that novel mutation tolerance and discovery, enabling target enrichment or hybridization capture to be possible even with degraded DNA, damaged DNA, highly divergent sequences. And key to conservation genetics allows probes to be designed from not exactly the same species as the one being studied. At Daicel Arbor Biosciences, again, our myBaits kits provide the reagents for this hybridization capture, either for custom probes designed for your particular targets of interest, as well as we have a range of pre-designed panels, such as the angiosperms 353 panel that I mentioned earlier, mitochondrial DNA panels also mentioned, and many more. And these can be applied to any hybridization capture workflow, regardless of whether you intend to use those at short or long read sequencing. We also have a full services NGS lab group and bioinformatics group, where our experts can take DNA that you've extracted, or in some cases samples themselves, and perform all the downstream steps of the workflow if that is of interest. So extraction, library preparation, optionally hybridization capture, sequencing, and analysis. Both our kits and our services group have well-established protocols that are compatible with a wide range of DNA sources and sample types, such as plant or animal tissues, for fresh or historical specimens, and much more. So finally, I just wanted to wrap up by highlighting, again, those some of those core specialties or strengths of Arbor's NGS team in our product and service portfolio. So whether your samples themselves are challenging or your targets are rare or complex, our team will work with you to find a solution that maximizes both your data recovery as well as your sequencing efficiency from a wide range of applications and sample types. So that's it. Thank you so much again to the ConsGen meeting for allowing us to be sponsors and for you to listening to this presentation. For those of you who are physically currently attending the ConsGen meeting, you should be able to find my card from our exhibit table that's there and also check out some of our product literature. Otherwise, please do check out our website and reach out to our team. We have a web form, we have a web chat, or our email, and you'll be put in contact with our team to start discussing project ideas or answer any technical questions that you have. So thank you again very much, and have a wonderful rest of your day.